Good evening, everyone. Welcome to another session on YouTube. We'll be discussing about a number of questions for the upcoming examination of June 2024. Just check whether the presentation is visible to you or not. Let's begin with the class now, right? So, before starting the video, before starting our class, if you're new to Botany Insider, please do not forget to hit the subscribe button. In addition, we have our June 2024 examination scheduled from 25th to 27th of July. So if any one of you who is interested or who is like preparing for this particular examination with the best of their ability, you can download our application and get yourself involved. We have a number of courses running for the students. One of the courses, the PYQ practice course, which is started or like which is for the June 2024 batch. In addition, we have a number of courses running for the same examination as well. In addition, for the December 2024, the examination which is going to held in the next cycle, we have already started off with a full course for the same. If any one of you who is willing to appear in the December 2024 examination, you can download our application and get yourself enrolled in the same. All the information for the same is in the description box, right? Let's begin with the class. The question is, a fly with apricot colored eye was crossed with sepia colored fly of opposite sex. In the F1, all the flies were wild type. The genes responsible for the two phenotypes were, or if you have a fly with apricot colored eye and you're crossing it with, like you have apricot colored eye and it is crossed with your sepia eye colored, then which is of opposite sex. So in the F1 generation, we saw all of them were the wild type, right? Question is asking you that the gene responsible for these two phenotypes were which of the following? First one is allelic, then you have non-allelic, then you have pseudo-allelic, then you have paralogous genes. Try the question, everyone. It's not very difficult. You have apricot colored and you have the sepia colored eye of the opposite sex, and you are crossing both of them together. So as a result of crossing each of these, question is asking you which are the gene responsible for the two phenotypes that we have. Try the question, everyone. Allelic, non-allelic, pseudo-allelic and paralogous. Try the question. Give it a try, everyone. Very good. Others also mark the answer. The two genes responsible for this particular phenotype are which of the following? Allelic, non-allelic, pseudo-allelic, and your paralogous. So, answer for this particular question will be that they were non-allelic, and because they were non-allelic, that's why there was normal segregation done, and because of the segregation you were able to see the wild type. That's why answer will be option number B, non-allelic, right? And not option number A. Remember about this particular information, everyone, that why or what was the gene responsible for the two different phenotypes that were there. The answer is non-allelic, because the genes were non-allelic. As a result of the same, we had the two different types. Even if they are crossing together, we have the wild type getting formed. Next question. Which of the following combination would best characterize the dominant phase of the life cycle? Question is, which of the following combination would best characterize the dominant phase of the life cycle? Try the question, everyone. Diploid gametophyte, haploid, diploid sporophyte, and haploid sporophyte. Which of the combination is the one that would characterize the dominant phase of the life cycle of pteridophyte? Try it out. Diploid gametophyte, haploid gametophyte, diploid sporophyte, and haploid sporophyte. Very simple question. Try it out. Very good. Others also mark the answer. Which of the following is a combination? Which of the following combination would best characterize the diploid phase of the life cycle of the pteridophyte? Very simple question. The answer for this particular question will be that it was a diploid sporophyte, right? Easy question. Answer is option number C. Next question. What is the pattern of cleavage observed in mammals? What is the pattern of cleavage observed in mammals? Radial, spiral, rotational, bilateral. What is the pattern of cleavage observed in mammals? Everyone mark the answer. Spiral, radial, rotational, bilateral. Very simple. It is rotational. So what's the pattern of cleavage observed? It is rotational. Next question. Which of the following regions of the target gene is not used for making RNAi construct to knock down its expression? Which of the following region of the target gene is not required or it is not used for making an RNAi construct to knock down the expression? Mark the answer for this. 5-UTR, 3-UTR, exonic and intronic. Try it out, everyone. Which region will you not be using for making up the RNAi construct? Very simple. Very good. Others also mark the answer. Simple and easy. Which of the region cannot be used or which of the target region will not be used for making the RNAi construct to knock down the expression? So the answer for this is the intronic region. You will not be having the introns, because introns are what? They are the non-coding sequences, right? So, because they are non-coding sequences, hence you are not going to use them in the expression formation, or you will not use them for the RNAi construct, because we have to knock down its expression. That's why intronic region is the correct answer for this one. D is the correct answer for this particular question. Simple question. D is the correct answer. Next, in which of the following stages of Arabidopsis thaliana, in the Arabidopsis embryogenesis, do the visible distinction between your adaxial and the abaxial tissue becomes initially apparent? In which of the following stages of your embryonic, in the Arabidopsis embryogenesis, do the visible distinction between adaxial and abaxial tissue of the cotyledon become initially apparent? Globular stage, zygotic stage, torpedo stage, and your mature stage. Try it out, everyone. In which of the following stages of Arabidopsis embryogenesis do the visible distinction between the adaxial and the abaxial tissue of cotyledon become initially apparent? Zygotic, embryonic, globular, zygotic, torpedo, and mature. Mark the answer, everyone. Very simple question. Answer for this is option number C. Torpedo stage is the correct answer for this particular question, right? This is very simple. Very good. Those of you got the correct answer. Next question. Try this. Mimicry where the deceptiveness of the mimic's signal is high and fitness consequences signaled to the receiver by the mimic is also high. Give it a try to the question, everyone. The mimicry where the deceptiveness of the mimic signal is high and the fitness consequences signaled to the receiver by the mimic is also high is Batesian, Mullerian, Fisherian, and your last one is Millerian. We need to find out which of the following or which of this mimicry is this one. So the answer for this is option number A. It's your Batesian mimicry. The mimicry where the deceptiveness of the signal is high and also the fitness consequence signaled by the receiver is also high. So in both of these scenarios, or The condition for both of this is option number A, that is Batesian. There are two major types which we study. One is the Batesian and the second one is Mullerian. So in the Batesian one, we have the both the things being high, mimic signal also and the fitness consequence also being higher, right? Okay, give it a try to this particular question. How many hydrogen bond involving the backbone of CO and NH can be observed in an alpha helix consisting of 15 amino acid residues? That is, how many hydrogen bonds involving the backbone of CO and NH can be observed in an alpha helix consisting of 15 amino acid residues, 10, 11, 12, and 13. In this, there's a specific formula that we apply for the number of H bonds. Try the question, everyone. How many hydrogen bonds involving the backbone of CO and NH can be observed in an alpha helix consisting 15 amino acid residues, 10, 11, 12, and 13. So in these type of questions where we have to solve the number of hydrogen bonds, that too in the alpha helix, if the number of residues is 15, like there's a formula for this, for the number of hydrogen bonds, it is n minus 4, because when we study the alpha helix structure, so in the alpha helix structure, there are in total, like there's a gap of 4 amino acid, first and fifth. Like the hydrogen bond formation occurs between the first amino acid and the fifth amino acid. So there's a gap of 4. That's why we take up the difference as 4, and as a result the formula of n minus 4 is taken into consideration. So number of hydrogen bonds is n minus 4. As a result, the answer is option number 11. 15 minus 4, that is 11 is the correct answer for this one. You just have to apply the formula over here and you'll get the answer for this question, right? Remember about this, how many hydrogen bonds are there. If it's alpha helix given to you, you just have to put the formula of n minus 4, that is 15 minus 4, which is 11, right? That's why B is the correct answer for this particular question. Easy and simple. Is the question clear to everyone? Yes, n minus 4 formula need to be applied in over here. Remember about this, right? One more question. Given below are list of morphological features and a group of plants that we have. So you have liverworts and mosses. In liverworts, like you have to find out which all morphological characteristics will be there in, and same for the mosses as well. Give it a try to the question, everyone. You have liverworts and mosses, unicellular rhizoids, multicellular rhizoids, presence of pyrenoids, stomata on the sporophyte and the dominant gametophyte. Which of the following options represents the correct matches between the two columns? Give it a try to the question, everyone. Last question for today. Mark the answer for the question, everyone. Liverworts and moss. So you have unicellular rhizoids, then multicellular rhizoids, presence of pyrenoids, stomata on the sporophyte, and the dominant gametophyte. Which of the following option represents the correct matches of the two things? So answer for this is option number D. That is, in the liverworts, we have unicellular rhizoids and your dominant gametophyte. In your mosses, you have multicellular rhizoids, pyrenoids, and your multicellular rhizoids, sporophyte, stomata on the sporophyte, and your dominant gametophyte. So D is the correct answer for this particular question, right? Remember this: liverworts, unicellular rhizoids, and your dominant gametophyte. In the mosses, you have multicellular rhizoids, stomata on the sporophyte, and the dominant gametophyte, right? So remember about this. D is the correct answer for this particular question. This particular question, this type of question has been asked number of times in the examination. So remember about this particular question and the entire classification of the bryophytes. It will help you mark the answers for the other topics as well. Okay, if any new topic gets asked in the examination, right? I hope all the questions are clear. From tomorrow, like from Monday, we'll discuss the topic wise, right? I'll take up a unit, some specific topics, and the questions will be discussed accordingly. So, if you're interested in joining in the question series concept, you in the topic series concept, you can join in the class from Monday, and we'll have classes mid like almost at the same time only because this is more comfortable. So let's meet in the next class now. Bye everyone. Take care.